Okay, let's do the last part of Carb Lecture 1. All right, so there's a couple things about this. Like one is how we make disaccharides. And the reaction that's used to make disaccharides is what's called a condensation reaction, just because it generates water. So there's a couple of key points here. One is those glycosidic bonds are covalent. Right, those atoms are sharing electrons, which in almost every case means it takes an enzyme to make the bond, it takes an enzyme to break it. And so when we look at this, for example, when this is combined together, when you have that glucose-glucose, you generate a water molecule. That's how you form that bond. And so now you end up with the glycosidic bond between two glucose molecules to make maltose. So what's probably more interesting for us is how we reverse that. And this is a hydrolysis reaction because in order to break that bond, the enzyme uses water. You ever seen this, a structure of this or anything? Of this actually, how the enzymes work? So imagine this enzyme, and what would we name this enzyme? It's going to break apart maltose. So here's maltase, which is a type of hydrolase, right? So maltase has a water molecule next to its active site, and it'll basically hold on to maltose and then use the water to break that glycosidic bond. So when maltose goes into our gut and maltase breaks it, that's how it's doing it. And now we have glucose-glucose that we can absorb. We'll go through all that in a few minutes. Good there? So that's pretty basic. Now let's go from oligosaccharides to polysaccharides. There's a couple of the more famous ones that we're going to talk about. And remember, polysaccharides are polymers of repeating monosaccharides. And for example, we have starch. It's a homopolysaccharide, which means it's all glucose. So it's all glucose, but the glucose molecules are oriented in two different types of structures. So starch has two inherent structures. And one of them is called amylose, and the other one is called amylopectin. Have you heard those terms? Right? One of them we say is the linear form, and the other one we say is the branched form. And glycosidic bonds and differences are why those structures are what they are. So here's amylose. Right, so you can think of it like amylose is the linear type of starch. It's all glucose, but look at the differences in the bonds. Glucose, 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 and this says et cetera, because these are long strings of glucose. The only glycosidic bond in amylose is alpha 1 to 4 glycosidic bonds. So right here's our anomeric carbon. It's downward, so alpha 1 to 4. So relatively straight across, and amylose actually forms kind of a wrapping structure, but it doesn't have any branch points. So we say it's unbranched starch. And then here is amylopectin. This is the branched form. So amylopectin has alpha 1 to 4. There's our alpha 1 to 4. And some of the glucose molecules have an additional glycosidic bond that is an alpha 1 to 6. That's a branch point. Remember all that? So what's more common in starch? Amylopectin. Probably 80-85% of starch tends to be amylopectin. Are there different types of starch? Absolutely. Different plants have, can have, so we can say, oh, it's starch from tapioca or corn or whatever. But in general, that plant will about 85% of its starch, amylopectin. Okay. Now here's where here's a good overview of this. So for example, each one of these little blue hexagons obviously is glucose. Here's our linear alpha 1 to 4. Here's our alpha 1 to 6. Here's starch unbranched form. So this is amylose. You can see these long chains of glucose. And here is amylopectin. See those branch points? Now, here's glycogen. Glycogen is pretty similar to amylopectin. Glycogen has alpha 1 to 4 and alpha 1 to 6. It's all glucose. Here's the difference. Glycogen has more branching than amylopectin. In glycogen, you tend to have an alpha 1 to 6 every 5 to 10 residues. In glycogen, you tend to have an alpha 1 to 6 every 5 to 10 glucose molecules. So a little gap there. Amylopectin, you'll have alpha 1 to 6 about every 20 to 25 glucose molecules. So more branching in glycogen. We'll talk about why that's important later on, but you can imagine glycogen is more dense because of that. And for metabolism, it actually means you can get glucose faster because of that. We'll talk about that. Here's cellulose. You've heard of cellulose, right? Cellulose is the most common organic molecule on Earth. It's all glucose. But we know that cellulose fits into what category of nutrients? It's a fiber. It's all glucose, but see the way its structure is different? Yeah, it's beta. So now if you look at it, here's all the glucose molecules. Here's glucose, glucose, glucose. So that's great, right? But they're beta bonds, right? So rather than being an alpha 1 to 4, it's a beta 1 to 4. Which means we can't hydrolyze that. But it's almost everywhere. It's in grass, it's in plant, it's in almost everywhere. It would completely change our lives if we could degrade cellulose. We could eat grass. I mean, we can, but we could get glucose from grass. But we don't. But we have bacteria, some bacteria that actually can. And we'll talk about that when we talk about fiber. But it's just that glycosidic bond. And remember, these enzymes are recognizing not only monosaccharides, but the orientation of that glycosidic bond. So amylase, the main enzyme we use to break down starch, can't hydrolyze cellulose.
Can you give any examples of heteropolysaccharides, or are those like man-made? Like, do any of them happen in nature?
There heteropolysaccharides. I'm blanking now, so no, I can't at the moment. But you do see them. You see some of them in nature. I'll think of one maybe. I'll get that to you.
Is the difference between soluble fiber and insoluble fiber, does that have to do with the bonds?
It can a little bit, but it's more about the chemical groups that are part of the fiber. So some of them are able to interact with water better than others. And so cellulose is actually an insoluble fiber. But some of them that are soluble fibers actually have a charge, like pectin or some of those. They'll have a charge that the water interacts with, and it's very basic. It's soluble fiber is just one that interacts with water and basically binds and interacts with water. So cellulose, you'd look at it, you'd think, oh, it's glucose, it's water soluble. But because it's so long and it tends to pack together, it's not water soluble. Do you need to draw the structures of cellulose, glycogen, amylose, amylopectin? Yep. And that'll be in the study guide. So you can just, and you can just feel free to say etc. Right? You can draw a few. Like you could draw 3 together. Okay, that's amylose. And then really, you can draw amylopectin, you can draw glycogen, right? So just practice on those a little bit. And then cellulose is the different one. Good there? Yep.
So if we were to draw like both like amylopectin and glycogen, glycogen.
Yeah.
Would we just like specify more branching for glycogen?
Probably wouldn't ask for both of those.
Okay.
Yeah, be honest with you.
Got it.
So let me find this one. Okay, lecture 2. So this is the outline for, we're going to talk about carbohydrate digestion, then we'll look at enzymes and their substrates, and then absorption of monosaccharides, and then we're going to kind of follow those monosaccharides through the different tissues, and that'll lead us into some of the metabolic pathways that we're going to discuss. And then towards the end of this, we'll talk about regulation of blood glucose, not today, but next time. We'll talk about type 1 versus type 2 diabetes pretty briefly on that. Okay, so one of the things that I mentioned before when we looked at that diagram, I said, in general, probably the most common type of carbohydrate that should be in most people's diets would probably be starch, right? It's a complex carbohydrate. And so one of the things we're going to look at is what are the differences in digestion between starch and disaccharides. And then we'll talk about the key enzyme for starch digestion, right? So now we're switching into this topic, and I don't have a better picture of this, so there we go. So this is carbohydrate digestion, and there's a couple of things to look at here. One is, here's the food source. This is the form of the carbohydrate in foods. And remember, starches have both amylose and amylopectin, and their digestion is different. But if we look down here, we have lactose and sucrose. So these are the most common forms of carbohydrates in diets. Now, obviously, if someone is eliminating dairy from their diet, then lactose is going to be pretty much eliminated. And then sucrose, I've got it at 30%. Okay? So for some people, that's a low amount. Okay? But if you look at the relative amounts there, those are just kind of ballpark estimates. But look at the way they're different. So if we go left to right here, let's look at starch. All right, we said it's amylose and amylopectin, and it goes into the gut, and it's got 2 phases to its digestion. Right? And phase 1 is luminal hydrolysis. In other words, when starch goes through the stomach and enters into the small intestine, when it's in that aqueous solution in the small intestine, digestion can start. Right? So that's just luminal. That's what's in that solution. That generates these molecules, maltose, maltotriose, and alpha-dextrins. We'll look at them in a second. And then next phase is actually on the brush border membrane. That's what BBM is, brush border membrane. See those 2 steps? Starch goes into the gut, it's in solution, digestion starts. Some of those digestion products now move to the surface of the intestinal cells, second step of digestion. So new location, new enzymes. Got that? That's what that means. We'll look at more detail in a minute. But we mentioned when we first started talking about digestion, we said, carbohydrate is absorbed as monosaccharides. So we've got to go from starch to glucose. And the complicating issue is there's 2 glycosidic bonds in starch. Here's lactose, right? Disaccharide. No luminal digestion, just on the surface. Can anyone name that enzyme? That's lactase, right? Apparently, I'm not very good at making it. I used to be. And in college, I realized, something changed. Better limit my milk intake. And we'll talk about that as we go through this, but that enzyme does that. And then here's sucrose. Its main enzyme, sucrase. It is on the brush border membrane. Okay? So guess what we're going to do? We're going to do a few things, one of which is we're going to talk about how those enzymes get to the brush border membrane. You all know, that's secretory pathway. That's the key to everything in life, right? Okay, so now let's use our imagination a little bit on these guys. Oh, sorry, let me go back. So now we're answering this question. What is a key enzyme for starch digestion? We know it's amylase. More specifically, pancreatic alpha amylase. Right, remember in your saliva there's some amylase? Helps a little bit, but that amylase goes in the stomach. Harsh environment, not a lot of activity. So then that starch goes from the stomach in the small intestine. And that's where the pancreas can allow its secretions to go into the small intestine. So it's pancreatic alpha amylase. That's the key one.
So in that case, would there already be alpha amylase like in the solution? So would it start in the phase 1? So like,
yeah, in the small intestine?
Yeah.
Yeah, so this is, yeah. So phase 1, luminal hydrolysis, pancreatic alpha amylase. See this? That's where it is. So phase 1 for starch is pancreatic alpha amylase. All right, so the starch is in a luminous small intestine. Pancreas is stimulated. Alpha amylase enters the small intestine. Starch digestion basically hits high speed or whatever you want to say at the high rate.
So does lactose and sucrose just go straight to the brush border?
Yeah, I don't know if I'd say, yeah, they're kind of mixing around in solution as they interact with the surface of the enterocytes. They can be digested. Yeah. We'll talk about lactase in a minute. Now, here's where it's interesting. More interesting than other stuff. So here's a diagram of pancreatic alpha amylase. This shape right here and these letters, A through E, indicate this is a shape showing pancreatic alpha amylase and its binding sites. So pancreatic alpha amylase has 5 binding sites. One cleavage point. In other words, so now imagine this enzyme is basically, this is the enzyme and these are the binding sites, right? So starch is going to be bound by the enzyme, and the enzyme is going to take 5 glucose molecules to fill its binding sites. And then the enzyme cuts once. So in other words, you got all of this in here, but only one of those glycosidic bonds is cut. And then what does the enzyme do? Releases. So here's why that's important. Alpha amylase only works at high efficiency when all 5 binding sites are filled. Alpha amylase only works with high efficiency when all 5 binding sites are filled. Right? And it has to do with affinity constants and enzymatics and stuff. But anyway, and alpha amylase only cleaves alpha 1 to 4 bonds. And it says that right there. Amylase only cleaves alpha 1 to 4 bonds. Now this is starch. So what's the other bond we have to digest? Alpha 1 to 6. Amylopectin is most of starch. Amylopectin has alpha 1 to 6. Pancreatic alpha amylase does not do that. So we need phase 2. So phase 2 is to complete starch digestion. Okay, but let's think about this. And what this means, obviously, is each one of these circles is a glucose molecule. Right, here's our alpha 1 to 4. N means repeating. 4 means, that's the open hydroxyl group. And here's number 1. See, let's get that diagonal line. That means that's the reducing end. So this is where we have the open anomeric carbon. But essentially all this is trying to show you is this is the linear portion of starch. Alpha amylase has to fill all binding sites, so it's looking for those linear portions. It's binding and it's cleaving and it's releasing. So here's what you end up with. You end up with maltose, maltotriose, and alpha limit dextrins. And I'll show you their structures in a minute. And alpha limit dextrins, also known as alpha dextrins or isomaltose, are 3 to 5 glucose molecules linked by both alpha 1 to 4 and alpha 1 to 6. Right? So because of that, the properties of alpha amylase, you don't end up with glucose. You end up with these smaller molecules in amylopectin, but you don't end up with glucose from this. So here's a diagram, and this isn't perfect, but hopefully this will kind of make this more easy to understand. So here's alpha amylase's activity on amylose and amylopectin. So here's amylose, for example, and this is repeating, right? So here's a straight portion of starch in amylose. Here's all our alpha 1 to 4. So you can imagine, amylase can bind right here, basically bind, fill its 5 binding sites, has high affinity for that, cleaves, and then you have maltose and maltotriose. 3 and 2 glucose molecules linked together with alpha 1 to 4. Here's the problem. Amylopectin has an alpha 1 to 6. You remember that term steric hindrance? That essentially means the structure of this molecule, that alpha 1 to 6, won't allow amylase to bind like this. It won't allow it to bind to this portion. It can bind here, it can bind here, but it can't bind across that alpha 1 to 6, which means it can digest close to that, but it's going to tend to leave these molecules that look like this. 5 glucose molecules with alpha 1 to 4 and alpha 1 to 6. That's alpha dextrins. So you end up with a lot of that. Products from amylopectin, such as alpha limit dextrins, represent about up to 1/3 of the mass of the final breakdown products of amylopectin. All right, remember this is phase 1. So you end up with a lot of this. So now what do you have to have? Well, now you have to have an enzyme that can degrade alpha 1 to 4 and alpha 1 to 6. Or a combination of enzymes. Okay, so this again, phase 1. And now let's look at what happens next. So phase 2 of carbohydrate digestion, these are hydrolases on the brush border membrane. They finish starch digestion. They also cleave the other major carbohydrate sources, lactose and sucrose. And these are commonly referred to as oligosaccharidases, if you've ever heard of that. And these are very important enzymes. We're going to talk about a few of them, mostly 3 of them, 4 of them, sorry. The next slide has a table. Oops. All right, so let's talk about sucrase, isomaltase, also known as alpha dextrinase, and maltase, and plus a galactosidase. Beta-galactosidase. So this table shows the enzyme and its type. In other words, enzymes 1, 2, and 3 here are 3 different alpha glucosidases. And you can imagine why they're called that. Their substrates have glucose and alpha glycosidic bonds. This enzyme down here, lactase, is a beta-galactosidase. Its substrate has galactose and a beta-glycosidic bond. So it's different than the other ones. And we can imagine the substrate for lactase, lactose. So let's talk about this really. This is probably the best point to talk about it, right? we've all heard of lactose intolerance, right? And lactose intolerance is basically because of a reduced expression of lactase. And that's the enzyme that breaks down lactose. So if you don't have that enzyme, you don't break down lactose. And so the lactose continues into the large intestine. And that's what can cause some of the side effects of lactose intolerance. So when that lactose goes into the large intestine, those bacteria love it. That's a significant energy source for them. So the bacteria can degrade that lactose. And as they degrade it, they'll make molecules that attract water. And so now water basically rushes into the large intestine, and that's why some of the side effects are bloating. Sometimes those bacteria will generate methane and other gases, so you end up with that. And also, you could use the term, sometimes it results in fecal urgency, right? And all of those things are caused because it's very common to have decreased expression of lactase as we get older. And why could that make sense? What do you think? If you think about like our life cycle or as we go through life,
Technically, you only need that enzyme when you're breastfeeding.
Yeah, so for newborns, right? So newborns, that's a major source of energy for them. It can be, and so when they have that lactose, newborns tend to be born with a full level of expression of lactase, even before we're born. During fetal development, lactase is being expressed, and so that way, when they're born, and that's their main fuel source, they can when they go on milk, they can degrade that. As we get older, it's very common that expression goes down, and there's kind of an RNA splicing mechanism that's temporarily regulated. In other words, as we get older, our cells tend to just decrease expression. It's very common to have lactose intolerance. I say this not because I'm defensive and I'm lactose intolerant, but because it probably in some genetic or ethnic groups, it's 80-90% of the adults are lactose intolerant. And essentially it means you still express some of the enzyme, just not as much as you used to. So there tends to be kind of a level of lactose that a person can basically handle. So when I first realized I was lactose intolerant, I thought I was allergic to milk or something, because this is a long time ago before they had any products, really. And so I got off milk for probably a year or two, and because I was drinking almost half gallon a day probably. So not a good time to drop your enzyme level. And I remember I was going through baseball practices, and pretty soon I couldn't make it through practices anymore because I'd go home for lunch and I'd eat a lot of food and a lot of milk, and then I'd get out on the field, and not very much longer after that I was in the restroom. Pretty soon the coach is like, "Where's Reeves?" And I thought, well, I don't know what's going on. And I thought, it's the milk. I'm allergic to it. And then later on, after I realized, oh, I'm not allergic to it. And then I kind of learned I can tolerate this much, but not more. So what we have to think about is, if you think about it, if a person eliminates milk and dairy products from their diet, that's a significant source of a lot of important nutrients. Calcium, high quality protein, and some other things that are good for us. So one of the things to think about is, can we continue to have milk products or dairy products in that situation? Unfortunately, milk that's treated with lactase is about 2 times as expensive. Like a gallon of that milk is probably $4 or $5. And so that can be an issue for a lot of people, unfortunately. So we have to realize, and then what else can you do? You can take pills. And the pills are essentially a recombinant or a modified form of the enzyme that goes through the stomach and remains active. So you take it with it, it goes through the stomach. They've modified the form of the enzyme so it handles the acidity, and then it goes in the gut and it's still active. So that's another way to do that. And certain food products obviously have more than others. So cheese and yogurts and stuff tend to have less lactose, so you can generally tolerate those better.
If you're continuously continuously consuming dairy, why would your body, like, wouldn't your body sense that and not decrease transcription of the lactase enzyme?
If that was the cue or the stimuli that the system is responding to, and it's not, it's actually age. Yeah, but good thinking. Yeah, that'd be great. Would save me a lot of money. But that's, yeah, so that's kind of what happens. So you want to be able to describe that to me and some of the side effects, and you'll see that in the study guide. I can put that in there. Right? So one thing to think about, that's the only enzyme that can do that. So if we lose that enzyme, we lose that ability to do that. There's another situation for that as well. So these enzymes like sucrase, isomaltase, and maltase are generally named based on their substrate. Sucrose is degraded by sucrase, right? And we know that's an alpha-1 beta-2. But look what else sucrase will degrade. It'll degrade sucrose. It'll also digest alpha-1 to 4 links of maltose and malto-oligosaccharides. So that enzyme can do 2 different glycosidic bonds. It's kind of unique. It actually likes this one better. I used to have the K catalytic constants on here, but if you're interested in that. This enzyme actually likes this bond better, but it's the only one that'll degrade sucrose, so we call it sucrase. And where are these enzymes? Brush border membrane. All these enzymes. They are on the brush border membrane and basically reach out into the lumen, and they wait for substrate. So they cleave their substrate, and then the monosaccharides that they can generate are absorbed. Next one, isomaltase. It degrades isomaltose, also known as alpha-dextrin, also known as alpha-limit dextrins. Those all mean the same thing. Remember, these molecules are 3 to 5 glucose molecules linked together by alpha-1 to 4 and alpha-1 to 6. Look what this enzyme does. It'll cleave both alpha-1 to 4 and alpha-1 to 6. So in your notes, you want to maybe indicate, put a star and go, isomaltase is the only enzyme that'll do the alpha-1 to 6. So that's a key enzyme. And then maltase, its substrate is maltose and malto-oligosaccharides. They both have the same glycosidic bond, right? It's just a matter of more glucose units, but it'll degrade alpha-1 to 4 links only. It won't degrade alpha-1 to 6. So it takes a combination of these enzymes at 2 different locations for carbohydrate digestion to occur. And we're going to look at how some of these enzymes get there, because this is a very, very interesting thing as well. And this has to do with the synthesis of sucrase and isomaltase. So now these 2 enzymes, sucrase and isomaltase, 2 different enzymes made from 1 gene. And I'll explain that in a second. So this is kind of a classic study that they did a long time ago in lab rats. And see these times? They were actually using this experiment to see how long it took these enzymes to move through the secretory pathway. That's why there's times here. So they did time course experiments. Okay, so bear with me. Let's look big picture. I know we're late. It's been a long week. Big picture, we're looking at the brush border membrane. So here's the brush border membrane. So this is a enterocyte. Now look where we are. This is some of the things we've talked about before. Here's the ER, Golgi, transport vesicle, right? This is secretory pathway. But look, it's only the end of it. In other words, we're not seeing the gene expression, we're not seeing the RNA, we're not seeing the first part of translation, we're just picking up here. So you want to be able to take this information and integrate it with the stuff you already know. So just looking at the end of it. This is the protein, the peptide, that lighter color. These little black lines represent glycosylation points. So this is where some carbohydrate is added. See what it says ER? In this case, some of the glycosylation is occurring in the RER. And then it continues in the Golgi. See the way we have this length of line here, and now it's longer. So this is further glycosylation here. And then look what it does. Right? It's packaged in the Golgi and then moves to the brush border, and it's embedded in the brush border membrane. So it's inserted in the brush border membrane. The protein is. So P is precursor. Precursor. And what is that? What do you think when you hear that? Is it active? It's not active yet. Precursor is an inactive form of the enzymes. So now look what happens. It goes from this to 2 molecules. And have you ever heard of trypsin? Trypsin is an enzyme that comes from the pancreas. It's a protease. So you can say trypsin is a protease from the pancreas. See where we're going? Post-insertional processing. It was inserted. Now it's going to be processed to become active. Trypsin cleaves the precursor. So trypsin cleaves the precursor into the 2 active enzymes. What? Sucrase and isomaltase. And you can't, I think maybe when I pulled this, I probably scanned this. Probably didn't have a digital form of this. This line right here should still be attached to the brush border. I love this textbook. The guy only did 1 edition of it. This is a nutritional biochemistry by Brody. I think he was at Berkeley when he did this. There should be a line here. So now look what happens. This protein is embedded or inserted in the membrane. Trypsin cuts it into 2 different enzymes. So you can imagine, one way to imagine it would be, let's say this is the enzyme, the precursor is stuck in the membrane, trypsin cuts it, so now it's exposing the active sites. That's the processing that activates it. So we say it's post-insertional processing. And now you have an active isomaltase, and S is sucrase. So a couple points. 1 gene, 2 proteins. Have you ever seen that before? That's kind of rare. How'd you get 2 proteins? Trypsin. So this is fair for me to ask you to go through this process from the gene. So you go through gene expression, you make 1 RNA, 1 mRNA, 1 protein that goes all the way to here, and end up with 2 different enzymes. So in other words, you need to be able to take the information that you already know and add it to the front of this. This is a great example. Don't just memorize the slide. You have to understand it. So you want to be able to go from gene expression to active enzymes for sucrase and isomaltase. Good? I think that shouldn't be hard. Everyone understand kind of big picture and all that? Yes?
So after the trypsin has broken it into I guess 2 enzymes.
Yeah.
So why is the isomaltase still embedded in the
They both are. They both are.
Oh, okay, that's what you're saying.
Yeah.
Okay.
So they are embedded, about 95% of the protein hangs into the lumen. About 5% anchors it. And that's for both of these guys. So about 5% of isomaltase is in the membrane, about 5% of sucrase is in the membrane. The rest is ready for substrate in the lumen. So now, for example, when isomaltose or sucrose interact with the enzyme, it'll cleave it, and it stays on that membrane.
Is there a specific gene that makes that precursor?
Yeah, so this gene is basically the sucrase isomaltase gene. Uh-huh.
You said 5%?
About 5% of the protein is embedded in the membrane. One more slide because this is just a summary. This is a good one to kind of go through and just see what we've talked about. Starches, 2 steps. Pancreatic alpha amylase generates these, and these enzymes finish it, so you end up with glucose. These disaccharides, just on the brush border, generates the monosaccharides that can be absorbed. Happens every time we have carbohydrate. Our next step in here is going to be, now for imagine these guys are in the lumen of the small intestine, then we're going to start following them through the different tissues. Not today, though. We good there? Any questions? Okay, guys. Have a good night. Have a good weekend.